Namaskar. I am Neeraj Kumar, Assistant Professor from Department of Zoology of Poddar International College, Jaipur. We are going to discuss about animal cloning. Introduction. What is the cloning? Cloning is the process of produce individual of an organism either naturally or artificially which are genetically identical. The term clone are coined by Herbert J. Weber. The cloning in biotechnology is the process of creating clones of an organisms and copies of cell or DNA fragments. It's called molecular cloning. In nature, organisms produce their own clones through asexual reproduction. There are no meiosis, that's why these are identical to each other. Types of cloning. The first one, gene cloning. Second, animal cloning. Third, therapeutic cloning. The first one is here, gene cloning. In gene cloning, many identical copies are formed by a single gene or its part. For this technique, use the PCR and the thermocycler. By this cloning technique, we can study the DNA more properly. Second one, reproductive cloning. By this technology, organisms are to be cloned. In this technology, three methods are used. First one, separation of blastomere. Second, Nucleus Transfer Technique. Third, Honolulu Technique. The first one is separation of blastomere. Blastomere are the separate from the donor animal. The cells of the blastomere are the totipotency in nature, mean these cells able to form complete embryonic structure. We can see here the development of larva from the zygote. The zygote is cleaved into 2-cell stage, then 4-cell stage, then form a complete larval body. If we separate it into 2-cell stage, then we can see here the only 2 larval body is formed, and if we separate it into 4-cell stage, then we see here the 4 larval body. Mean it's proved each blastomere is the responsible for development of embryonic structure. Second, nucleus transfer technique. Nucleus transfer technique is used for the producing clone animal. The nucleus transfer technique is categorized into the various steps. First, remove the nucleus from the recipient cell. The selected unfertilized egg cell, the first treated with the colchicine and cytocalcicin. Due to the cytoskeleton get immobile and remove the nucleus by the enucleation pipette. We can understand by this diagram. The recipient egg is holded by the holding pipette and the nucleus is removed by the enucleation pipette. Next, select the donor cell. Which cell have to be cloned? It's called the donor cell. Example, embryonic stem cell. The Roslin Institute produced the Dolly ship through nucleus transfer technique. They select the fertilized egg, and this fertilized egg they cleaved into the blastomeric condition. In the blastomeric condition, the blue cell are known as ICM. Mean these are the inner cell mass which are separated out and cultured in the medium, and this is known as the embryonic stem cells. These embryonic stem cells are transferred into the enucleated egg cell. This process is called nucleus transfer. The third process is nucleus transfer. In this, donated nucleus is transferred to the recipient cell or egg. The fusion and the start cleavage of both substances by the electric shock, Sendai virus, and maybe the enucleation pipette. We can understand by the diagram. Here the egg cell and its chromosome are removed, and here the rest is here only plasma. And from the donor adult cell we select the nucleus and fuse with the electric shock of both of them, and they start their cleavage. After the complete the cleavage, they form the blastomere, and these blastomere are ready to implant with the uterus wall. This process is called implantation. Fourth point is implantation. The fused cell is the implant at the blastomeric stage into the mature female uterus. After the implantation, the embryonic development occur and the development into the fetus. After the gestation period, the baby is produced. It's called a clone. How the Dolly ship was produced? The Dolly was born on the 5th July 1996. Roslin Institute, Scotland, by the Ian Wilmut and their team. They select the Scottish blackface and the Finn Dorset sheep. From the Scottish blackface sheep, they select the egg cell and remove its nucleus by the enucleation method, and from the pin dorsal chip they select the memory cell and fuse them with the electric shock and start their cleavage. After the complete the cleavage, they form the blastomeric condition, and this blastomere are ready to implant in the surrogate Eve uterus wall. And after the implantation, there the embryonic development complete, and after the gestation period, the surrogate Eve produced a baby lamb, which named was the Dolly. Next technique is Honolulu. The technique is developed by the Teruhiko Wakayama, a technique that uses the adult cell and the nuclei for the cloning animal. The first clone is called Cumulina. In this technique is modified technique of the Wilmut and the Gordon's animal cloning technique. By this technique we can produce the 3 generation of mice. How the Honolulu technique was worked? From the one mice we select the oocyte, and from the another mice we select the somatic cell or nucleus from the donor. And from the selected oocyte of the mice we remove its nucleus, and from the another mice we select the normal somatic cell and take out from them nucleus, and the nucleus and from the another mouse and the enucleated egg cell are fused with the electric shock and the enucleated pipette, and after 1 to 2 hours their chromosome get condensed, and after the condensation of the chromosome they form pseudo pronuclear formation. After the formation of the pronuclear body, it start cleavage after the 24 to 76 hours, and after the complete structure formation of blastomere, it implant into the surrogate mother or mice. After the implantation, the surrogate mother produce the mice. These are known as cloned mice. Third one is therapeutic cloning. This technology is useful in the organ transplant technique. This technique is avoid the graft rejection. What is the graft rejection? Graft rejection is an immunological destruction of the transplanted tissue or organ between the 2 members of the strains of a species. If we solve it by therapeutic cloning. If a patient have the organ failure, then we take out from its body a biopsy cell and enucleated human oocyte is the implanted by the biopsy cell and formed it a blastula, and these blastula are cultured and form the embryonic stem cell, and after embryonic stem cell in the in vitro differentiation get and the implant into the human or patient body. What is the strategy of the therapeutic cloning? This from the diagram we can understand. If a patient had the organ failure or any other problem related to organ, we can transplant the organ from another body. How it's work? From the patient we take the somatic cell and the biopsy, and from the we select the enucleated oocyte and transfer it into and fused with the electric shock, and the blastomere condition get, and these blastomere condition are the embryonic stem cell, and mean these cell can form any type of the other cell, mean neuron, hepatocyst, renal cell, cardiomyocytes, pancreatic isolates, hematopoietic cells, any kind of cell, and we can trigger them and the implant into the patient body, and we can solve the problem of the patient related to their organ failure. Thank you.